in this video we'll be completing with the local anesthetics part 1. The local anesthetics chapter is divided in two parts. This is the part 1 of the video and we'll be uploading part 2 soon. Okay, so you have to watch in order to understand the whole topic. So here specifically we will be talking about the SAR of local anesthetic and the benzoic acid derivatives drugs in detail, and rest we will be also doing with the aminobenzoic acid derivatives today, and the lidocaine and the miscellaneous will be completed in the part 2 of the video. So let's start with the basic information that you have also studied in the your HAP or pathophysiology subjects. Okay, so let's start and understand what is local anesthetic. Local anesthetic is nothing but an agent or a drug that is either given topically or parenterally at a general localized area that produces the local anesthesia by reversibly blocking the nerve conductances that transmit the feeling of pain from the locus to the brain. This simply means it is a reversible suppression of any kind of pain, okay, or loss of sensation without the loss of consciousness. This is very important definition. The term anesthesia is defined as a loss of sensation without any loss of consciousness. Okay, this local anesthetic decreases the excitability of the nerve cells without affecting the resting potential. And because of the action potential, the ability of nerve cells to be excited seems to be associated with the movement of sodium ion. So in the mechanism which we'll see for local anesthetic, there is simply a blockade of sodium channel passage, okay, and due to which there is a anesthesia like effect produced, okay, which stops the transmittance of any kind of movement or touch sensations created to the human body. Okay, so you can see the mechanism of action of local anesthetic is believed to be via the sodium channel blocking effects. Okay, the local anesthetic drug binds to the channel in the area just beyond the selectivity filter or P region. So simply what happens, the local anesthetic goes and drug binds to the area just beyond the selectivity filter and P region. Okay, and due to this, the local anesthetic binds with the sodium, binds to the receptor and blocks the sodium ion passage into the cell and blocks the formation and propagation of action potential, and this blocks the transmittance of message of pain or even touch from getting to the brain. So simply in the mechanism we have to understand key in the local anesthesia effect, it is potentially seen or observed that there is the blocking of sodium ion channels. So what happens when the local anesthetic binds to the receptor, it simply when it binds to the receptor, it simply it binds to the area or the area, it binds simply to the channel or the area beyond the selectivity filter or the P region, and then thereby blocking the passage of the sodium ion, and thus that blocks the formation and propagation of action potential. This blocks the transmittance of message of pain or even touch from getting to the brain. The ability of local anesthetics to block action potential depends on the ability of the drug to penetrate and to the tissue surrounding the targeted nerve as the ability of the drug to access on the binding site of the sodium channel. So simply क्या अगर किसी ने पूछा what is the mechanism, so you can write this कि ये जो local anesthesia है, anesthetic है, ये bind करता है to the channel of the beyond the P region or the selectivity filter. Second point you will write it blocks the sodium ion passage into the cell and thereby blocks the formation and propagation of action potential, and this leads to the blocking of transmittance of message of pain or touch from getting to the brain. Okay, then we have simple diagrammatic study. Okay, it is triggered by mechanical stimulation, thermal stimulation, chemical stimulation, bradykinin or histamine like autopoids, okay, which are sensed by the receptors in the brain. Okay, so there are free nerve endings which due to which we can sensitize any kind of stimulation, and thereby we feel the pain. Okay, so I'll not cover this. This is for just a basic knowledge what happens, how the channel happens. These are the cation channels. Okay, simply the action potential takes place when there is an exchange of sodium channel. So there is simply a blockade for entering of the sodium channel on the receptors, and as there is a blockade, the action potential is also stopped, or the propagation of formation is also blocked, and hence there we can by pain the we cannot feel the pain, but we are conscious. Okay, so we have the here simply voltage gated sodium channel. What happens, it is all the mechanisms I need not be completing here. You can just study by looking in the reference. It is just a reference slide. Okay. Local anesthetic do not access the binding site by entering into the sodium channel from the exterior of the neuron. The molecules are too big to pass by the selectivity filter. If local anesthetics do not access the binding site via external site, how do they get the site of action? You have been wondering. So, the experimental evidences or the clinical trials have been shown certain that local anesthetic must access the binding site via the hydrophobic or the hydrophilic pathways. Okay, so this was generally simple question which comes in the mind, and the answer is for that is here. Okay, then here there is again a reference slide which is telling you all that how the anesthetic passed in the uncharged form into the hydrophobic pathway, and how in the hydrophilic pathway they enter inside the brain. As we know ki the molecules are very large, so it is just an elaborative way, okay, how it enters. Okay, so we start with a very important slide of today, that is the SAR, or the structural activity relationship of the local anesthetics. Now, this is generally the general structure of the local anesthetics. Sorry. This is the aromatic ring, okay, or the lipophilic ring, which has a linkage of ester or amide. Okay, so this is the linker, and here we have the amino ring or the amine ring. So we'll just see and study about how are the actions of the general structure. Okay, so this is the general structural formula. So let's study. Yeah, I'm sorry, I had the diagram, I just don't remember it. Okay, so this is the first ring, that is the aromatic ring. Then we have the linker and the amine group. Okay, so the structure of the most local anesthetic agents, as I said, consists of three parts as shown above. The first, the lipophilic ring or the aromatic ring, that is maybe or substituted. The linker, which consists of ester or amide, that can be of various lengths, okay, and the amine group, that usually is the tertiary amine, and the pKa between is 7.5 to 9.0. So the aromatic ring, so what the points to be remembered for the aromatic ring? Now in the aromatic ring, this aromatic ring adds the lipophilicity to the anesthetic, and that helps the molecule to penetrate to the biological membrane. So as the name itself, it is having the aromatic ring or the lipophilic ring, so it simply adds lipophilicity and enables the molecule to penetrate through the biological membranes. Now it is also thought to have the direct contact with the local anesthetic binding sites on the sodium channel. Okay, the aromatic ring is believed to interact with the local anesthetic binding sites with the pi-pi interaction or pi-cation interaction with the S6 domain component of the sodium channel. Why? Because we know in the mechanism there is a blockade of sodium channel. So the sodium channel is playing an important role here. So we have seen the 3 points. First कि जो लोग aromatic ring है वो lipophilicity ऐड करती है जिसकी वजह से मॉलिक्यूल इजीली बायोलॉजिकल मेम्ब्रेन या फिर ब्लड ब्रेन बैरियर को इजीली पेनिट्रेट कर सकता है कोई भी बायोलॉजिकल मेम्ब्रेन को. Then second we have seen कि it has a direct contact with the receptor binding site. Okay, or receptor binding site on the sodium channel or local anesthetic binding site on the sodium channel. Then it is believed to be binding to the local anesthetic binding site with either pi-pi interaction or pi-cation interaction with the HS6 domain component of the sodium channel. Now, this SAR study of para-substituted ester type of local anesthetics showed that the lipophilic substituents and electron donating substituents in the para position increase the anesthetic activity. SAR studies have shown कि जो भी para substituents of local anesthetics है, especially the ester type ones and the electron donating substituents. Please remember it is electron donating substituents, okay, in the para position increase the local anesthetic activity. The lipophilic substituents are thought to be increase the ability of molecule to the nerve membrane and increase the receptor site. So whatever the lipophilic substituents are binding to the local anesthetic site, they are not only uh, you know having the ability of the molecule to penetrate the biological membrane, but also to the increase of the affinity of the receptor binding site. Okay, एंड एज वी सी की इलेक्ट्रॉन डोनेटिंग ग्रुप या फिर लोकल लिपोफिलिक सब्सटीट्यूशन रहेगा ईस्टर टाइप का लोकल एनेस्थेटिक का पैरा पोजीशन पे स्पेसिफिकली, तो वो एनेस्थेटिक एक्टिविटी बढ़ा रहा है. बट अगर इलेक्ट्रॉन डोनेटिंग ग्रुप्स रहेंगे वो क्रिएट करेंगे रेजोनेंस इफेक्ट बिटवीन द कार्बोनिल ग्रुप एंड द ring. Okay, and they, so what will they do, this electron cloud of the oxygen, so did the increases the affinity of the molecule with the receptor. मतलब क्या हो रहा है, सिंपली इलेक्ट्रॉन डोनेटिंग ग्रुप्स अगर प्रेजेंट है रिंग पे तो वो क्रिएट करेंगे रेजोनेंस इफेक्ट ऑफ द कार्बोनिल ग्रुप विच विल रिजल्ट इनटू द इलेक्ट्रॉन क्लाउड इंक्रीज एंड दिस की वजह से एफिनिटी ऑफ द मॉलिक्यूल is also increased. ठीक है, when the aromatic ring substituents is substituted with the electron withdrawing group, the carbonyl oxygen decreases and the anesthetic activity decreases as well. ठीक है, so we have what we have seen for the local anesthetic activity कि सबसे पहला जो था कि लिपोफिलिक रिंग या फिर एरोमेटिक रिंग जो है हमारी, ओके, फर्स्ट पॉइंट वाज दैट इज वो ऐड कर एरोमेटिक रिंग जो थी वो लिपोफिलिक एसिटी ऐड कर रही है जिसकी वजह से लोकल एनेस्थेटिक एक्टिविटी जो है सॉरी जिसकी वजह से हमारा पेनिट्रेशन हो रहा है मॉलिक्यूल का इनटू द बायोलॉजिकल मेम्ब्रेन. ठीक है, देन द सेकेंड पॉइंट हम लोगों ने देखा वाज जो भी लिपोफिलिक रिंग जो भी बायोलॉजिकल मेम्ब्रेन होता है लिपोफिलिक रिंग वो उसको डायरेक्ट कांटेक्ट होता है लोकल एनेस्थेटिक बाइंडिंग साइट से, ओके, ऑन द सोडियम चैनल. फिर जो लोकल एनेस्थेटिक बाइंडिंग साइट को बाइंड करती है उसका इंटरेक्शन इधर पाई पाई इंटरेक्शन होता है या पाइकेटाइन इंटरेक्शन होता है ऑफ द S6 डोमेन कंपोनेंट ऑफ द सोडियम चैनल. SAR studies of para reveal that जो para position के इलेक्ट्रॉन डोनेटिंग सब्सिट्यूएंट्स है और इलेक्ट्रॉन और लिपोफिलिक सब्सिट्यूएंट्स है पैरा पोजीशन पे वो इंक्रीज करते हैं एनेस्थेटिक एक्टिविटी को, और जो भी इलेक्ट्रॉन डोनेटिंग ग्रुप्स होते हैं वो एनेस्थेटिक एक्टिविटी को इंक्रीज करते हैं रिसेप्टर क्लाउड की वजह से, और प्लस वो एफिनिटी ऑफ मॉलिक्यूल भी इंक्रीज करते हैं. ठीक है, एंड जो इलेक्ट्रॉन विड्रॉइंग ग्रुप्स होते हैं उनका इलेक्ट्रॉन क्लाउड जो होता है कार्बोनिल ऑक्सीजन के वजह पास वो डिक्रीज हो जाता है जिसकी वजह से एनेस्थेटिक एक्टिविटी भी डिक्रीज होती है. देन वी हैव द लिंकर. अब जो लिंकर है, द लिंकर इज यूजुअली ईस्टर और अमाइड ग्रुप ओके और ये अलग अलग लेंथ का हो सकता है. सो अलोंग विद द हाइड्रोफोबिक चेन ऑफ वेरियस लेंथ्स. ओके जो लिंकर है वो जनरली ईस्टर या अमाइड होता है एंड दैट कैन कंटेन a hydrophobic chain of various lengths. Number of carbon atoms in the linker is increased and the lipid solubility, protein binding, duration of action and toxicity also increases. मतलब पहला पॉइंट इसमें ये है कि जो लिंकर है वो या तो ईस्टर या तो अमाइड होना चाहिए, उसके साथ हाइड्रोफोबिक चेन होनी चाहिए वेरियस लेंथ की. जैसे नंबर ऑफ कार्बन इंक्रीज होंगे, प्रोटीन बाइंडिंग, लिपिड सॉल्युबिलिटी, ड्यूरेशन ऑफ एक्शन, टॉक्सिसिटी भी इंक्रीज होगी. अब जो ईस्टर्स एंड अमाइड्स है ये दोनों ही बायो आइसोस्टर्स है एक दूसरे के. दैट इज दे हैव सिमिलर साइज, शेप एंड इलेक्ट्रॉनिक कैरेक्टर्स बट दे डिफर इन द स्टेबिलिटी ईदर इन विवो और इन विट्रो. ओके एंड द अमाइड्स जो है वो ईस्टर से बहुत ज़्यादा स्टेबल है बिकॉज दे हैव द लॉन्गर हाफ लाइफ दैन द ईस्टर. Okay, the nature of the substituents of the aromatic ring can affect the also confer a steric block to protect the linker from the metabolism, and thus the binding affinity and stability of the anesthetic molecules is affected, and the linker as well as the functional group of the aromatic ring. यानी जो नेचर ऑफ सब्स्टिट्यूएंट्स होता है एरोमेटिक रिंग का, मतलब यहाँ हम लोग लिंकर की बात कर रहे हैं बट नेचर ऑफ एरोमेटिक रिंग भी बहुत ज़रूरी है बिकॉज इट कैन इफेक्ट द electronic nature of the linker and can contribute to the drug potency and stability. ओके द सब्स्टिट्यूएंट्स ऑफ द एरोमेटिक रिंग जो है ये कन्फर करते हैं एक स्टेरिक ब्लॉक टू प्रोटेक्ट द linker from metabolism, and thus the binding affinity and stability of the local anesthetic molecule is affected by the linker and as well as the functional groups. Okay, then the first ester local type anesthetic ester local anesthetic ester type was synthesized was procaine, that is novocaine, in 905. The para-aminobenzoic acid or the PABA metabolite of common ester class of drug is believed to have a responsible for allergic conditions, and some patients have experienced it with the local anesthesia. इसमें हम लोग को ये पॉइंट्स याद रखना है सबसे पहले कि जो भी ईस्टर या अमाइड लिंकर जो है ये वो ईस्टर या अमाइड कंटेन करता है अलॉन्ग विद द हाइड्रोफोबिक चेन, ठीक है, ऑफ वेरियस लेंथ. देन सेकेंड पॉइंट याद रखना है हमें कि जो भी नंबर ऑफ कार्बन एटम्स जब भी इंक्रीज होगा, लिपिड सॉल्युबिलिटी, प्रोटीन बाइंडिंग, टॉक्सिसिटी, ड्यूरेशन ऑफ एक्शन भी बढ़ेगा. अमाइड और ईस्टर्स एक दूसरे के बायोआइसोस्टर्स है, यानी दे हैव सेम शेप साइज एंड इलेक्ट्रॉनिक कैरेक्टर्स बट जो डिफरेंट करता है दैट इज स्टेबिलिटी इन विवो और इन विट्रो. देन हम लोगों को याद रखना है कि जो नेचर ऑफ एरोमेटिक रिंग है वो बहुत ज़्यादा इंपॉर्टेंट है बिकॉज द इलेक्ट्रॉनिक नेचर ऑफ लिंकर को अफेक्ट कर सकता है एंड विच कॉन्ट्रीब्यूट टू द ड्रग्स पोटेंसी एंड स्टेबिलिटी. अब जो सब्स्टिट्यूएंट्स होते हैं एरोमेटिक रिंग की वो कन्फर करते हैं एक स्टीरिक ब्लॉक टू प्रोटेक्ट द लिंकर फ्रॉम द मेटाबॉलिज्म, जिसकी वजह से बाइंडिंग एफिनिटी और स्टेबिलिटी अफेक्ट हो जाती है एज वेल एज द लिंकर का फंक्शनल ग्रुप. ठीक है, और उसके बाद जो हमने फर्स्ट ईस्टर टाइप का नोवाकेन लोकल एनेस्थेटिक सिंथेसाइज किया था वाज नोवोकेन और प्रोकेन इन 905. और इसका जो इम्पोर्टेंट मेजर मेटाबोलाइट है पाबा वो रिस्पॉन्सिबल है मेजर एलर्जिक कंडीशंस के लिए इन द लो पेशेंट्स who are experiencing local anesthetics. Then we have the last one, that is the amino or the nitrogen or the amine ring. अब जैसे कि मैं बोली थी कि ये जो लोकल एनेस्थेटिक्स होते हैं जिन अमाइन रिंग का, ये टर्चरी नाइट्रोजन होते हैं मेजरली, और उनका पीके होता है बिटवीन 7.5 to 9.5, और देयरफॉर ये फिजियोलॉजिकल पीएच पे बहुत ज़्यादा केटोनिक एंड न्यूट्रल फॉर्म में मॉलिक्यूल दिखता है. ठीक है, क्या है पहला पॉइंट कि ये लोकल एनेस्थेटिक टर्शरी नाइट्रोजन कंटेन करते हैं जिसका पीके 7.5 से 9.5 है और ये फिजियोलॉजिकल पीएच पे कैटायनिक और न्यूट्रल फॉर्म में मॉलिक्यूल एग्जिट कर। अब ये फिजियोलॉजिकल पीएच पे जो भी अनआयोनाइज्ड फॉर्म है लोकल एनेस्थेटिक का वो कैलकुलेट कर सकते हैं विद अ वेरी वेल नोन इक्वेशन, हैंडरसन हैजलबैच इक्वेशन, दैट इज पीएच पीके प्लस लॉग बी अपॉन पीएच। और ये जो लोकल एनेस्थेटिक कंपाउंड्स जो बाइंड करते हैं एनेस्थेटिक रिसेप्टर को, दे आर मोस्टली इन द आयोनाइज्ड फॉर्म। ठीक है, यू हैव टू रिमेंबर दिस। टॉक्सिसिटी एंड साइड इफेक्ट्स बहुत इंपॉर्टेंट है लोकल एनेस्थेटिक का। एक मेजर टॉक्सिक साइड इफेक्ट है दैट इज मिथेमोग्लोबिनीमिया। ठीक है, cyanosis as a result of the formation of methylglobinemia may occur after the administration of local anesthetics like lidocaine, prelocaine and benzocaine. Okay, so ये methanoglobinema कंडीशन में ऐसा हो जाता है कि जो हमारा हेमोग्लोबिन है वो जब आयन हेम ग्रुप से कंबाइन होता है तो वो जनरली फेरस आयन प्रोड्यूस करता है, बट जब लोकल एनेस्थेटिक देते हैं एक पर्सन को तो वो आयन हेम ग्रुप के साथ रिएक्ट करके फेरिक एज फेरिक देता है। ठीक है, सो ये फेरिक आयन के वजह से जो हेमोग्लोबिन फेरिक आयन कंटेन करता है इसके वजह से द ऑक्सीडाइज्ड, अब ये ऑक्सीडाइज्ड फॉर्म में जो आयन है ये ऑक्सीजन को नहीं बाइंड कर पा रहा है। ठीक है, और उसके वजह से एज यू नो ऑक्सीजन और ब्लड साथ में कैरी करते हैं। ओके, एंड दिस ऑक्सीजनेटेड ब्लड इज नथिंग बट द हेमोग्लोबिन। तो उसके वजह से जो इंसान के फिंगर्स होते हैं और डिजिट्स होते हैं दे द नेल्स टर्न्स दिस ब्लू। ओके, एंड इसका ट्रीटमेंट है 1% मिथिलीन ब्लू सोल्यूशन। ओके, ये साइनोसिस को रिवर्स करने के लिए, दैट इज मिथेमोग्लोबिनिया को रिवर्स करने से साइनोसिस रिजल्ट एंड नॉट रिस्पॉन्ड, बट ये ट्रीटमेंट को 100% रिस्पॉन्ड नहीं करता। ओके, पेशेंट विद इंक्रीज रिस्क फैक्टर्स फॉर डेवलपिंग ड्रग इंड्यूस मिथेनोग्लोबिया। अब ये किनको होता है तो द चिल्ड्रन और द किड्स दैट आर younger than 2 years, the geriatric patient, okay, और the anemic patients और जिनको genetic deficiency है और 6 glucose 6 phosphate dehydrogenase और nicotinamide adenine nucleotide। ठीक है, तो ये एक एक्सेसिव डोज की वजह से हो जाता है और उसका ट्रीटमेंट है इनवेनस इन्फ्यूजन ऑफ 1% मिथिलीन ब्लू सोल्यूशन 1 mg per kg ओवर द वेट बॉडी। ठीक है, so side effects and toxicity एक वो हो गया। then we also have like excitable membranes जहाँ पर sodium and potassium channels in heart, nicotinic acetylcholines and nerve cells in the CNA face the side effects and toxicity. That is, one we have in general neuromuscular junctions and CNS are more susceptible to, and then the cardiovascular system for this toxic effects of local anesthetics. Okay, and in this primary effect you can also experience certain severe CNS depressions or convulsions. Okay, and obviously जो local anesthetic का जो प्रोकेन है उसका मेटाबोलाइट था पाबा, उसको वजह से हम लोग का एलर्जिक कंडीशंस भी होते हैं, सो यू हैव टू रिमेंबर स्पेशली द ईस्टर टाइम। ओके, then this is again for a reference slide what are the difference between local anesthetics and other anesthetic drugs, okay, which you might have covered in your HAP. So we'll start with the again important topic, benzoic acid derivatives, the first class of local anesthetics. First drug is cocaine. This was first used for topical anesthesia and it was isolated from the coca leaves of the native people of Andes Mountains, chewed for multiple times including local anesthesia properties to ward off the fatigue. Yani what happened? There were the people from Andes Mountains. I'll tell you in the short this history behind this. So there was There were people or tribes from the Andes mountains. They used to work very hard, okay, and due to the working, they used to find these leaves or trees. So what they used to do, they used to just pluck one leaf and chew it. And they observed, or scientists observed, after chewing, or chemists observed that after working, the crystals of that coca leaves would numb their tongues, and they would anesthetize their cornea during the they would anesthetize that area and become numb. There was no sensation or loss of pain for few hours. Okay, then the German scientist or surgeon in 1884 came into the picture, who demonstrated a successful use of cocaine to anesthetize the cornea during the eye surgery. And then it was one of the most prominent surgeons at Johns Hopkins University. Then they read about the investigations, and unfortunately, the doctors who were in the research got addicted to it. So today, the cocaine that is used as a topical anesthesia for mucous membranes, okay, for using 4 to 10% of solution. If the solution remains for membrane for 5 minutes, anesthesia and vasoconstriction of the area will occur, okay, and this can inherent vasoconstrictor properties requires no additional epinephrine. Okay, so this is the structure. Here you have the atropine ring, and the rest of this is the ester linker, okay, amide linker, and this is nothing but the aromatic or lipophilic ring. Okay, so this is the tropine moiety, aromatic moiety, and the okay, and the cocaine results in the vasoconstrictor properties, ability to inhibit the catecholamine, excitation, dysphoria, tremor, seizure activity, hypertension, cardia, myocardial ischemia, and infarction are all the major side effects. Okay, and this is generally the metabolism. Japan hydrolysis, okay, you get ecogenine, methanol, and benzoic acid. Then we have the next drug, hexaikine. It is a short-acting local anesthetic, inhibits sodium and channel conduction. Overdose can lead to headache, tetanus, numbness, tingling around the mouth and trunk. Sometimes convulsions also occur, and inability to breathe and decrease heart function. Okay, so this is the general structure of the of your local anesthetics, and just you can compare. Okay, so this is nothing but your hexylcaine, and this is nothing but a meprylcaine. Okay, this is meprylcaine. Relative potent inhibitory action. Okay, you can just see the difference is here it is having one CH3, and here it is having an extra CH3. Okay, then piperocane, you just have a piperazine ring extra, whereas the structure is same. And even in the cyclomethazine, you can see there are cyclic ring, okay, and the rest you can compare the structure, it is similar. Then we have amino benzoic acid derivatives, in which the first drug is benzocaine. It is a local anesthetic because it does not contain a tertiary amine. The pK of the aromatic amine is 3.5, ensuring that benzocaine is uncharged physiological pH. Okay, this is just a difference. Rest the ring is same. Okay, so you are having a 1, 2, 3, and 4 butyl ring. Okay, butyl or butane you are having. And benzocaine is generally used for endoscopy, bronchoscopy, topical anesthesia, and again its toxicity is resulting in methemoglobinemia. Okay, and infants and children are more susceptible to this because it has been reported that the benzocaine lubrication of the endotracheal tubes used in the endoscopy is also used to treat a painful diaper rash. That is a multi-purpose use. Okay? Then we have the next drug, that is the procaine. Okay, that is from the aminobenzoic acid derivatives. It was synthesized in 904, which was addresses the instability of cocaine and local irritation it produces. The procaine जो है, it is very fast metabolized into PABA, paraminobenzoic acid, and acetaldehyde. Yes, paraminobenzoic acid only. Sorry. And okay, you can see here, which in the presence of cholinesterase in the liver via ester hydrolysis by a pseudocholinesterase, and the elimination half-life is approximately 60 seconds. And as we know कि पाबा is a major metabolite, okay, active metabolite. इसकी वजह से हम लोगों को we are getting allergic conditions or allergic reactions, okay. And these are also used in the peripheral nerve block in the epidural agent to diagnose the pain syndromes. Okay. The rest drugs are butane, propoxycane, tetracaine, and benoxycane. So the general structural formula of for the ester remains same, and what are the differences? Just you have to study that. And from lidocaine onwards, we'll be completing in the second part of the video. Thank you, everyone.